think it's better to be here. So we have chapters 4, 5, and 6. This chapter, we're going to learn more specifically about bacteria and archaea, which are not bacteria, but are similar to bacteria. This is not going to be a hard chapter. I'm wondering maybe all the hard stuff is in the very end, which Dr. Jackson, when I mean hard, I mean more physiology, where you're getting into transcription, translation, conjugation. Here we're just going over basically anatomy and function, which is interesting because you'll get to see how some of this relates to pathogenicity, which you'll see in your field. So today we're going to just do half of this. We're going to get up to 4.3. So we're basically going to look at the big structures of bacteria, what size and shapes, and then these external structures. I really encourage you when you're studying, draw it out. Use different colors. It's amazing how much your brain remembers things from pictures and colors and visually, or I should say physically moving your hand or verbally talking it out. Even if you just have a little whiteboard at home that you draw it out on and erase it, that's a really easy way to study. I bet you can find coloring books for this. That's what we need. We need coloring. That's how kids learn. I'm telling you, I have toddlers. I'm like, how do they learn everything so fast? Because they're physically doing everything. I'm racking my brain. I'm like, how can I teach genetics with crayons? Can I do this? How do I do it? Okay. So this is bacterial form and function for our first section. Okay. So if we take bacteria, archaea, and eukaryotes, how are they the same? How are they a little bit different? The biggest division is eukaryotes have a nucleus. We know that. And before, remember, we used to call everybody else a prokaryote before the nucleus. It wasn't until recently that we looked at archaea and said, we were doing genetic analysis, and we said, oh, wait a minute, these things are not bacteria. They are really weird because they have some aspects of eukaryotes and some of bacteria. So now we got to throw our whole system out. So now we're going to say there's bacteria, there's these archaea, and then everybody else over here with a nucleus is a eukaryote. So bacteria and archaea just have their nuclear mass floating around in the cytoplasm. Most of them have a cell wall, like 99% of them have a cell wall. For bacteria, it's some kind of form of peptidoglycan, and there are always exceptions to this rule. So I'm going to tell you later, like here's this bacteria that has sterols instead of peptidoglycan. But archaea, they don't really have peptidoglycan, and there are some cell walls in eukaryotes, like a fungi has chitin, right? And a plant has cellulose. They don't have that either. So they have their own unique makeup for their cell wall. And then internal structures, eukaryote cells, like your cells have things like these lysosomes, peroxisomes, all these membrane-bound organelles, and bacteria and archaea do not have those. Much simpler structure. This looks like it's a lot, but it's really not. What's important for you to know, what makes, like every single cell has to have, when we say what is a living cell, they have these things. So this is specific in all bacteria, they have this. Okay? Even if it's an archaea classification, you have to have what we would call, in this textbook, they call it a cell membrane. In biology, maybe they called it a plasma membrane. Do you guys remember the lipid bilayer? So whether you want to call it a cytoplasmic membrane, a cell membrane, a plasma membrane, that is all the same thing. It's kind of like if you considered a water balloon, that would be the balloon, right? They have to have some kind of nucleic acid. So it could be DNA, it could be RNA, but in our bacteria and archaea, it's just sort of this mass, okay, just sort of sitting in there. They have to have ribosomes. Okay? These are not considered membrane-bound organelles because they don't have a membrane. It's ribosomal RNA and a protein stuck together. That's what these little dots are. Okay? We're going to talk about those on Thursday, but just as a reminder, ribosomes are what make protein. So if you have DNA, but you don't have ribosomes, you don't have any protein, you're not a functioning unit. That's a virus, right? You're not able to do what you need to do. And then cytoplasm. Cytoplasm, for this class, we're just going to say it's basically the watery interior of the cell. We'll get into more about cytoplasm versus cytosol on Thursday, but it's basically the watery interior of the cell. So they all have to have that. That you should know. Like if I'm a bacteria or an archaea, I have to have these things. Other bacteria have some of these other structures that we'll get into. S layer, I'm not going to go through them now because we're going to go in detail, but fimbriae, an outer membrane. Most have a cell wall. Some of them even have cytoskeletons. This is a newer discovery. Okay, a pilus, that's for a reproductive structure, capsules, inclusions, microcompartments. And this class is kind of cool because I have seen it, this shows my age, over time, how things have really changed in this field as we have better technology and we can find things. And it's really neat. Like, we didn't have microcompartments and granules when I was taking this class. Plasmids, we'll talk a lot about those. Flagellum for motility, and then endospores and intracellular membranes. So the big ones, again, what do they all have to have? For the rest of those, you'll just have to understand what they are necessarily. Okay. This is a good organization page that I would star as kind of your reference when you're studying. Because this goes, okay, in order of, if you're on the outside of this, we're just going to say bacteria to keep it simple. If you're on the outside of the bacteria and you're moving into the inside, and I just started to make a little chart out here, we're going to start on our surface layers. So we're going to talk about things like the flagella for locomotion, and then we're going to get into surface layers. Remember, some of them have them, some of them don't. And then we get into the cell envelope, which are these three structures, and then the inside is all the organelles. Okay, so that's the whole design of the first part of the chapter. So if you have an idea of that, then that helps you learn the information a little better. Okay, so that's where we're getting to. First, bacteria, they can do everything, which is cool. Okay, so you are a multicellular organism, and you have storage, and you can slowly digest, and you can think and reproduce and have emotions, but they can reproduce, they can metabolize, they can make and break down nutrients, and they are one single cell. That's really cool. The other very cool thing that they do is they can group up and link together and act like a completely different organism. So the way they act when they're single is very different when they act when they're multicellular. If you want to think of them, they behave differently as one unit. When I say behave differently, their genetic profile changes. They can't change their DNA sequence, but there's something called the epigenome. Epi means above. This is what turns your genes on and off. If we looked at your epigenomic profile, we could look at you and say, what genes are you making a protein and which genes are shut off? It changes when they're single versus in a group, right, which is really cool. So they act differently. They can have different properties. And they might be called a colony, or what you guys would be more concerned with is biofilms, because that's a problem in the medical field. So these colonies, when I was saying, this is something that they really recently discovered is something called nanowires. It's literally a filament of metal that's connecting these cells, and they send things to one another, like electrons. They can put a substance and send it. This is like the internet for, bugs. How do they do this? So they apparently came up with this before we did. And it has them all sort of cooperating together, just like your cells in your heart can beat together by sending an electrical current. So this is really cool stuff. Imagine where we're going to be in another 10 years. Right, anybody in here have recurrent ear infections as a kid or know somebody that had them? Yeah, this is your page. So they used to just give you an antibiotic, right? And give you an antibiotic and give you an antibiotic. And then they're like, man, we've given this kid 15 antibiotics. This is not working. And then they discovered biofilms. So what they realized was, this is your inner ear. If you zoom in, this is your tympanic membrane, which is your eardrum right here. Okay? And then here is this pocket in here. It would fill up with fluid. This is your eustachian tube. This is when you're trying to swallow on the airplane and it pops your ears and equalizes your ears. Well, this becomes inflamed and closes up. This comes with fluid. And then you have these terrible, terrible air aches. You have all kinds of throat problems, as you in the room know that have this. So they can't treat this with antibiotics. So what they said now is, we're not going to give kids antibiotics. This is a really common procedure. How many of you know somebody with tubes in their ears? Like kids, they get them done all the time. This is what it looks like. They actually put a hole right here in the eardrum, and they insert this little piece of plastic tube in there. And all it does is drain this fluid out. So they're not actually stopping the infections. What they're doing is just letting it drain out. So what they realize, what the infection are, is these biofilms. So it's like multiple layers of bacteria, and one antibiotic isn't going to do it. 2 antibiotics isn't going to do it. In fact, we can't come up with an antibiotic soup that kills these things. So it's better to do this and just let it drain out. So they insert the tube, they push it in, so it's facing in here. And as that child grows, the eardrum starts to close back up that hole, and it twists the tube and twists the tube until the tube pops out into this part of the ear, and it's real tiny. Normally, it just sits in there in a little bit of wax, and the doctor checks the ears every 4 months to see if the tube popped out or if it's still in. And then eventually this closes up, and it heals. There may be complications, but that's essentially what they do now for kids. So if you suffered through all those, sorry, you didn't get tubes. Okay. So basic arrangements and size, and no, you do not have to memorize these details here. You should know the average size of bacteria is 1 micron or 1 micrometer. Those are the same terms. Okay, so that's about the average size of a bacteria. What I'm showing you here is some really crazy things. I think somebody had, maybe this person was drinking when they named this, Thio Margarita. I don't know. This one is huge. It's the biggest one that has been discovered, and it can be seen by the naked eye. So just give you an idea on the scale. I mean, you cannot see E. coli, right? And here's what an E. coli would look like next to this. So this is like a 0.5 millimeter, these big bacteria. Okay. On the other side of that, mycoplasma used to be the smallest bacteria that came in, very, very tiny. But now they have found nanobacteria. So if they just find something smaller than we already have, we call it nano. I don't know what's going to be the next level down. I feel like in science, we just keep finding things. The better the technology, oh, here's something even smaller. Remember when we thought the nucleus was just electrons and protons? Yeah. Now we're like, whoa, string theory, what? Okay. So nanobacteria, even smaller. 3 basic shapes, and you need to know this for lab too, so this will be easy for you. Coccus is basically a sphere. Okay, bacillus is a rod, and then spirillum is a spiral or corkscrew. Okay, within that, because bacteria are really able to adapt to their environments, we say they have pleomorphism, okay, which morph is shape. It just means there's a lot of varieties. You guys could all be the same strain of a coccus bacteria, but some of you might be a perfect sphere, some of you might be an oval, some might have a bean shape, some of you might have a pointy head, okay, even though you're the same. Or you might be the exact same, it just depends. But there's all different types of coccus. It just basically think of a circle, and here's some examples of what they might look like. This almost looks like popcorn kernel. Okay, so that's coccus. Bacillus, basically a rod. Another one that goes in this grouping is Vibrio. Okay, so this would be all the different types of rods, bacilli. But here's Vibrio. It looks almost like a comma. It's kind of curved. Okay. Spirillum, I'll show you those. There's 2 basic spirillum. This one, okay, which is kind of like a helix. And then this one is called a spirochete. It's more like a corkscrew. Okay. These ones, when we talk about the arrangements, we're going to talk about coccus and bacilli like to group up. These ones don't do it as much. Spirilla can sometimes do chains, okay, but spirochetes usually don't. They're kind of cool looking, right? Okay. Here's the arrangements of the cocci, and I see that this is covering up word. I think that just says of. Okay. If there's 2, know your word roots. This will help you if you know your Greek and Latin. Diplococci, okay, that's just 2. Tetrads are in 4. The ones that are important for you guys to know are staph and strep. Okay, right? Because you've heard of this. You're like, staphylo, strepto, like what does that mean? That is an actual arrangement of a specific type of bacteria. So if it's staphylococci, they look like grapes, like a cluster. If it's strep, they're in a line. Okay, it looks like beads on a string. Sarcina is this little structure here. If it looks like a cube of 8, 16, or more. I'm not going to have you look at pictures, but I would definitely ask you. The reason why I say you should know strepto and staphylococci is because those are big major ones in your field. I always say everyone's going to be a nurse, right? Is that what you're going into? Does anyone take this class that's not in nursing? I just assume. I don't know. Okay, good. I'm not going to say. Okay, for bacilli, they can be the same thing. Diplo, 2. Okay, strepto. You already know what strep means, right? It's a line. So that'll be, and then they have something weird called palisades, okay, which it kind of looks like this end to end. So they're joined up, but like this. Now we're going to do these external structures. Okay. So the first outer layer that we're going to talk about is if you have something stuck on you, like your appendages would be your limbs, your arms and your legs, right? So this is like the arms and legs of your bacteria. We have flagella or an axial filament. We're just going to call them basically, well, I'll show you the difference, but that's motility, so they can move around. They do move around in the body, fimbriae and pili. Okay, so those are for attachment. So a lot of times bacteria are going to attach to your, say, epithelial cells, and then they colonize, and then they start to reproduce, and then they make you sick. Pili can have channels between the bacteria. Okay, so let's start with our flagella. Okay, this is blurry, but this is to show you kind of, if you got really close up. I mean, look at this structure. It almost looks like somebody in Greece made this. Okay, this is what it really looks like, right? You think, oh, that's the cartoon. No, that is what it looks like. This is the filament. This would be the whip part. Here's a hook, and then here's the basal body, and this is attached right here through this cell envelope. So that's really cool, and it whips around. So it can be attached at the end of a cell. That's called polar. Okay, so if it's on just polar on one end, it's monotrichous. If it's lophotrichous, that means it's polar on one end, but it's got a tuft of hair. Okay, so here would be mono. Here's the tuft, lopho. Amphi means on either end. So it's still polar because it's on the end. Okay, and then peritrichous means all over the place. So peritrichous is not polar because it's not on the ends. Peri means around. Does that make sense? Okay. If you know your word roots, this makes your life just a lot easier. If you're bilingual, this makes your life a lot easier because you see it, right? A lot of you probably pick up on the words. Okay, so flagella is for motility, and we can use this for identification. We mentioned this in some other chapter, but we can't see flagella. If we're looking at a microscope, it's just so hard. They're so small, especially on a bacteria. We could do it, but it's harder. So instead, what they'll do usually is inoculate something, like here's a tube, and I just threw this picture in this morning just to show you what it would look like. You'll do a motility assay where you inoculate one of your bacteria, and then you can actually see this strip. That's the bacteria moving and growing down. So this would be semi-solid. You could do that hanging drop technique. Remember where it has a little concave part in your slide and you put a drop of water? Would have to be big. So why are they moving around? Your bacteria might be doing something called chemotaxis, which you're going to learn if you take immunology. That's a big part of cells moving towards something. Usually when we say chemo, think chemical. It's moving towards a chemical signal. So that means it could be a positive signal. Maybe it's a nutrient. That would be towards. If something is positive, I'm going towards it. So for you guys, if I serve you lunch or donuts, you're going to come up. The chemical signal would be the aromas coming in and linking to your receptors in your nose. Negative, if I brought something like broccoli in. Like, no thanks, I'm good. You'd move away from that. Okay, so say it's something that's a harsh chemical, then they're going to go away from it. And they have receptors just like you do, but it's on their flagellar membrane, so it can actually propel them towards or away from wherever they want to go. When they're going straight, and they're actually moving forward, or if you want to think of it away, we call that a run. So it's a counterclockwise motion of their flagella, and it's a linear direction. It's going to look like this. This straight line. This is in green is a run. A lot of times, though, they're not moving in a straight line because they're picking up signals as they go. So they're going to do a run, and then they're going to stop and do what's called a tumble. That's when they reverse their motion in their tail, and it causes them to stop, and then they'll change, and then they'll go counterclockwise again, and then go again. Okay, so a phototaxis is specifically if they move towards light. Okay, so here would be one that's going no stimulus. It's just kind of going and make it a run. It tumbled, run, tumble, run, tumble. The R is run, T is tumble. But this is one if it's moving towards a stimulus. It looks kind of erratic, but remember it's picking up chemical signals and then going, oh, I got to go this way. It's like the GPS isn't really working. It's going to go in and out. A periplasmic flagella is kind of the same thing, but when we've been looking at, they've just been free form. This is one that's actually within two membranes. So it's going to be actually enclosed between the cell wall and the cell membrane. So we don't have that up there yet, but the cell membrane, remember that's a plasma membrane, and the cell wall is on the outside. So it's going to be in between there, and it looks like this is kind of wrapped around. This is a cool picture because you can actually see one when they did the processing of this specimen. It released it. So you can actually see it pop out. It wouldn't normally look like that. It would be wrapped around like this. That's what they look like. So it's actually enclosed, and then it does its motion and it moves it. So that's the difference between a flagella and an axial filament. We'll see these usually in spirochetes, the little corkscrew ones. These structures are more for attachment. You don't want to move. You want to attach and colonize and reproduce. So a pilus or the pili, those ones we're going to see for conjugation, which I'll talk about. Fimbriae or fimbria, these ones are just for adhesion. It just helps them stick. Sometimes flagella can help them stick, but not really. I mean, mostly they're just locomotion. Okay, so let's talk about these fimbriae. These are just like little bristles all over. Most of them have protein, and this helps stick. So this might be one of the things that helps the formation of biofilms. What scientists are trying to look at is what kind of protein structures are in here. Is it a little different than what we have? Because if it is, that's a target for an antibiotic. And we're trying to get savvy with our antibiotics because they evolve much faster than we do. E. coli, gonorrhea, likes to use their fimbriae to attach to our epithelial cells. So here's some E. coli in the intestine. This is what they might look like on an electron microscope. Kind of looks like hair. This is a good hair day. I know it looks bad. The pili, this is also known as a sex pilus because it is used for reproduction. This is so cool. This might be one of my favorite things in science because it's like, this is so cool that they can do this. Okay. Instead of actually reproducing, like sexually reproducing, where they're going to come together and mix up their genes, okay, or cloning with asexual, they actually form this pili structure, which is a tube. One forms it to the other and attaches, and then it just sends over genetic material. So it would be like me copying some of my genes and saying, here you go, you want some blue eyes? There you go, have some blue eyes. Isn't that crazy? So in their case, they'll be like, hey, you want some antibiotic resistance? Here you go. I've got one of those. I've got 6 of those. What do you got? So that's conjugation. That's really cool. We do this in laboratories sometimes. If we want to put something into a cell, we can kind of shock it, and it opens up some of these pores, and we can try to get stuff into our bacterial cell. Okay, but we'll have, I'm always like, I don't know if you're learning that. I think you are. I think it's in chapter 9. I don't know if you got chapter 9. Okay, so those are the appendages. So that I didn't put up there, but it would be obviously on the outside. Okay, surface coatings. These are the ones that not all bacteria have these. So the first one is going to be S layer, and it's just called S layer. I know you can't really see this, but that says S layer. For you 3 people that can read the board. S layer is cool. It's like armor. If you've ever seen chainmail, maybe you've been to that medieval times. This is what it looks like. Okay, they take these proteins and link them together, and there's all different structures that can do it. It's actually really beautiful. Right? And they only are going to produce this if it's in a hostile environment where they need to protect themselves. Like maybe it's too acidic or too basic or too hot or too cold or has sulfur or methane or whatever it is that they don't like. But, so they can form this all the way on the outside. The glycocalyx, we've mentioned this one. This one is also repeating, but this is a sugar polysaccharide units. If you're not familiar with your biological molecules still, I would go back and review that from the first test because you really have to know the difference between a carbohydrate and a protein and a lipid when you get into all of this protocols for your lab work and then for all these structures. Okay, there's 2 different types of glycocalyx. You can have the slime layer or you can have the capsule. You're not going to have both. So the slime layer, be careful. That is not the S layer. Those are 2 different things. Okay, so you're not going to have both of these. You're going to have one or the other. This is glycocalyx, which is basically your big carbohydrates just wrapped around the cell. Okay, so it's going to prevent water loss, nutrient loss. The capsule is more for protection from things like white blood cells. Okay, this is definitely a tighter pack. Okay, so this would be slime layer and the capsule. The capsule is really important because right here would be the cell wall, okay, and the plasma membrane and anything else, let's say. And it's going to have all these proteins and receptors that are specific for bacteria sticking off of it. So what this capsule does effectively is wrap around and hide all those. So when these little bacteria are bumping into your white blood cells, when the white blood cell goes to attach to a receptor, it gets nothing. So it can't see the bacteria, and then it can replicate and then take over your body and make you sick. Okay, here's what the capsule might look like, a little in there. And then if you have this on a plate, it almost looks like you blew your nose on there. Yeah, gross. So if it looks slimy, gross, you'd be like, aha, capsule. So capsules are formed by pathogenic bacteria. Why are they pathogenic? Because they make you sick, because they can hide from your immune cells, your white blood cells. Part of the glycocalyx, this is another structure that can help form biofilms. Specifically, we know capsules and the glycocalyx can be part of the teeth, the bacteria that get stuck on your teeth, which is hopefully not being stuck on your teeth. But also, what's for a medical community would be things like a heart valve and IUD. Okay, so different devices like plastics and things that they put into the body. They're trying, if you're in bioengineering, to really find a surface that they can have that a bacteria can't colonize. Good luck, right? They live in volcanoes. They can live on anything. These are some of the ones that I was telling you where when they get together, they act differently. So this is really wild. You might have this bacteria that is susceptible to penicillin, but then it gets in a biofilm, and now it's no longer susceptible to penicillin because what it has done is changes its epigenome. So the genes are still the same. It's just the profile, which ones are turned on and off, is different, and it now has resistance. So that's a little scary. So that was our surface layers. Now we're going into the cell envelope, which is 3 sections. I'm going to go outer to inner. You might have an outer membrane. And I'll put a star because not only some bacteria have that. Then you have the cell wall. And then you have the plasma membrane. Okay, so that's going out to in. Okay. The cell membrane is the plasma membrane. I call it a plasma membrane. They call it a cell membrane. The cell wall is peptidoglycan. And if you're like, what the heck is that? Remember, that's a big sugar with proteins attached into it. So these 3 layers can act as just one unit together. This is what the outer membrane would look like. I put a little star by this because only some bacteria have that. Most of them, though, like 99% of them have that cell wall, and then they all have the cell membrane. So we have to talk about the Gram stain because this is where they began to differentiate between the outer membrane and the cell wall in some of these bacteria. So more detail on this. Remember, this was cool because this was developed back in 1884. We think we're smarter than those people back then, right? We are not. In fact, we're probably stupider because they had to figure things out and think. Okay, so we've actually not grown more intelligent as a species. They had it down pretty good. We just have more technology now. Well, it's the same method that Hans Christian Gram used. We still use this. Okay? And I don't know when you guys are going to do this stain, but I remember positive is purple and negative is pink. And it's all because some of them have that outer membrane and some do not. So if you're Gram positive, you're purple, you only have a cell wall in the plasma membrane. There's no outer membrane. So this is non-existent. Okay, so there's only 2 parts. If you're Gram negative, you have that outer membrane. So be thinking of it like, that like a protective shield that you don't absorb the Gram stain. Okay, it's negative because it can't get through. That's a lot of like lipids, and it can't absorb it. Okay. So this is what they would look like. So this big, thick section, that's a bunch of proteins. That's your peptidoglycan. This is the plasma membrane. Okay, so this is gram-positive, and it can soak up that gram stain. Over here, if you're gram-negative, you have a very thin cell wall. But then you have this thick outer membrane. The outer membrane is essentially just another lipid bilayer. It's just like the cell membrane down here, okay, but it's obviously a different composition. Okay, so this is negative and this is positive. I think that's actually a really cool picture. So again, if you draw this, especially with color, that'll help you remember. Okay, so let's talk about the cell wall. We're going to talk cell wall, cell membrane, and then come back to that outer membrane. The cell wall can shape your bacteria and its structural support, okay, due to osmotic pressure. So let's talk about this in case you don't remember. Here's your bacteria cell. It's got a lot of solutes inside. Okay, let's say that it is 3 molar. You're like, what the heck does that mean? Remember moles per liter? Okay, so that tells you how many solutes. And let's say that it's only 1 molar solution out here. That means there's more solutes out here than in here. Water always flows from high to low concentration. There's more water out here. So water is constantly going to flow in here. What would happen, this bacterial cell would explode. So the way to prevent that is having a cell wall. So if you have that cell wall, even though water is filling up and water is going to push out on here, you're going to have that thick cell wall so that it doesn't explode. We don't have a cell wall, and our cells will explode. But we're multicellular, and we can control that a little better than they can. So cell walls are made of peptidoglycan. That's the basic structure. We're going to see there are some exceptions, but like I was saying, like 99% of them have peptidoglycan. So it's sugar with these little peptides attaching it. It's flexible, okay, but strong. So this is what it might look like. These little circles are your sugars, and these yellow strips are the proteins, these amino acids. It's just a couple amino acids, so we say a peptide. Okay? And no, you don't have to memorize this, but this is cool to see it, that here's a sugar molecule, and here's your amino acids connected to another sugar. And you might actually see some of this, like N-acetylglucosamine, uramic acid. These are some of the things that trigger your immune system and say, oh, this is not me, because we don't have those structures. Okay, so more details about the cell wall for gram-positive. It's really thick. You do not need to know these numbers, but it just gives you a reference. It's like 20 to 80 nanometers. Okay, it's got acidic polysaccharides. Okay, so things like teichoic acid or lipoteichoic acid that are embedded within all these proteins, these peptidoglycan. So those acids are important. It helps maintain your cell wall. It helps them enlarge quickly for cell division. Okay? And then it provides this acidic charge on the cell surface, which helps when they're attaching in your body. Gram-negative cell wall is this little pink strip down here. Much, much, much thinner, only 1 to 3 nanometers compared to 20 to 80. You see the difference? We say it's somewhat rigid, but compared to a gram-positive, it's much more flexible because it's very thin. Okay, but it has this outer membrane for protection. It is more sensitive to lysis, which means this exploding, because this isn't as rigid to protect it. Now here's all the weird things. If you're not gram-positive or gram-negative, some of them are something in between, right, that. So we'd say they don't really have completely positive or negative. Some do not have a cell wall. Those are the really weird ones. Some of them might have something called mycolic acid. Another name for mycolic acid is cord factor. I'm just going to call it mycolic acid, but I put that in there in case your lab book calls it something else. They would be in Mycobacterium, Nocardia. They're really long fatty acids. So be thinking back to what we already learned. I think it was in chapter 2. A fatty acid, remember, is a hydrocarbon with a carboxyl group. Do you guys remember that from the fat molecules? Okay, and that makes these little bacteria pathogenic. We can't use the gram stain for these because obviously it's not going to work because they don't have peptidoglycan. So you're going to use an acid-fast stain. So this one you should know. Acid-fast is, if you're not going to be testing these, don't worry. Tuberculosis or leprosy, we don't use those in lab. Here's some TB, work with that. But that's what they would do in a laboratory to test for this. It's called acid fast. It's not a fast test where you're like, oh, this is so fast. They mean it, think of it like fastening, because the dye holds fast. It means tight, like it grips very tightly to the bacteria. There's a strong attachment. That's what the word comes from. There's so many lipids in there, it's waxy, so it's not going to hold on to an alcohol stain. Okay, so you've got to use an acidic stain to grip onto it. Where are you guys at in lab? I'm curious. What are you doing? What? You already had a practical? I feel like it just started. So you know all about gram stains. Good. You guys can come up here and teach me. Okay. The mycoplasms, they do not have a cell wall, okay, but they have sterols. And I don't know if we went into sterols too much, but that's like a wax. That's all what that is. So they're resistant to lysis because they have this really waxy coating. These are the ones that are really tiny. If you work in a laboratory, mycoplasms like to contaminate everything. And they are a pain in the butt to get rid of. So you'll know if you have it because you'll be like, what is, my cells look funky. And then when somebody has it, it's like having an STD. Who has gonorrhea? Who has mycoplasm? I remember the few times when I worked in a lab that somebody brought it in, right? Somebody contaminated it. And they'd be like, who has it? Oh, did you use their media? And like it was like gossip in the corner about who had mycoplasm. So you don't want to be that guy. You need sterile technique. Okay, these ones are known to be highly pleomorphic. They don't all look the same. So even though they're in the same colony, they might have really different shapes, like even donuts, which is fun. Okay, so this is all the same bacterial strain, but you're seeing all the different kinds of shapes in here. Okay, now we're going to talk about this outer membrane. Okay, so this is only in gram negative. The big part of this is lipopolysaccharide, LPS. You guys have to know that. It's such a big trigger for the immune system. My PhD is in immunotoxicology, so I studied how environmental contaminants affect your immune system. We would use LPS to get our cells activated. We would hit them with a shot of LPS and get them all ramped up and then do our assay. So this is something that your immune cells know to attack. It's a big marker. It's considered an endotoxin. It produces septic shock in our body if we have too much of it. So it stimulates fever, septic shock. So what's going to happen, and I think I might have mentioned this one, if you have a bunch of this bacteria and it dies rapidly in your body and releases all this LPS from the outer membrane into your bloodstream, you have a systemic-wide reaction with all your immune cells releasing this chemokine and these cytokines that open up all your vessels. They dilate, and you lose your blood pressure rapidly. Then you have no blood going to any of your organs. So you can die pretty rapidly. So that's bad. So we definitely have to be careful with gram-negative infections. Okay? And a lot of these, there's these called lipoproteins. They're going to attach that outer membrane to this little tiny cell wall. So they're going to be right in between here. So think about that, a lipoprotein. That's a protein with a little fat stuck on it. So here's your lipoproteins down here connecting. Okay, here's your outer membrane, which is essentially, it's just like a plasma membrane on the outside. It's made of phospholipids. Here's your LPS molecules. They have these pores. It's just called a porin. It's essentially just a protein that's going to go all the way through, so it can control what's coming in and out of the cell. Can also be used as a defense against certain antibiotics. So if you're working at a clinic or and you're in the hospital, they're going to want to know what kind of infection you have, if it's gram-negative or gram-positive. But you know what's fun? You just go to the doctor and they just give you tetracycline or doxycycline or penicillin, right? Just the basics. And when that doesn't work, that's when they're like, uh-oh. Okay, we don't have a very fast test for this where we're going to say you have gram-negative or positive. This is usually like if you have something funky and they can't figure it out and Dr. House is busy, they've got to send this off to a lab, which is going to take a while, and then hopefully you don't die in the process. Hold on a minute, don't die. We've got to get our lab results back before we treat you. Right, so that's something that we definitely could solve a lot of problems if we had a quick diagnostic test to know what kind of bacteria or if you had a virus. Okay, so this outer membrane, It helps protect against antimicrobial chemicals and makes them harder to kill than a gram-positive. So even though gram-positive had that big peptidoglycan layer, we know how to attack that. We have antibiotics that break those cross-links apart really easily, as long as they're not resistant. So we have all those, but we're not as good as getting that outer membrane. And so we definitely have to treat these a little bit differently. The cell membrane is just the plasma membrane, which you've learned in basic biology, right? That's just a lipid, phospholipid bilayer. It's somewhere like 30 to 40% phospholipids and 60, 70% proteins. I'm not going to ask you the exact numbers, right? As long as you understand what it is. Again, remember, mycoplasms are a little weird because they have a bunch of sterols. They have wax. And then archaea are really weird. They have hydrocarbons. They don't have fatty acids, but so they just have the chains of hydrocarbons and no carboxyl group. Cell membrane is really important, and you've learned this in, like I said, your other biology class. For what the difference is, think about for us, we have mitochondria where we make ATP. That's where we do cellular respiration, right, with the double membrane and the matrix and all that. They don't have mitochondria, so how are they making ATP? A lot of times what they'll do, some of them have more than one membrane in there that they can use, but sometimes they'll go between, right here, this plasma membrane and the cell wall, and they'll set up that hydrogen gradient, and they'll make ATP that way. They can make nutrients using the cell wall, and then, of course, transport, getting things in, getting waste out. We say that cell membranes are selectively permeable. Okay, so if it was just permeable, that means anything can come in and out. Like our doors are permeable. Anyone can walk through those doors and walk out, right? If we select, though, what if I say, oh, no, you can only come into my classroom if you have a ticket, and I'm only giving my students ticket, then I'm selecting who can get through the door. That's what it's like, except for in this case, they're going to do it by charge and size. It's all biochemistry. Okay, so if you have the right charge or the right size, you can get through. It's going to select out based on other things. Water, gases can usually get through really rapidly, but if you're a charged molecule, you get stuck. Phospholipids have charges, and they've got all sorts of negatives and positives up here. So what happens if you're a charged molecule, you get stuck by the charge interaction. Right? You might get through eventually if you're small, like water is charged, but it gets through slowly enough that it's not a problem. If they want to have water come in, they can have a special transport to get it in and out quickly. Secretion, that's when you're actually secreting something out of the cell. Remember, extracellular is outside, intracellular means within. So this is, when we talk about the cell envelope, that's all three of these. So it just depends on if you're referring to a gram-negative or positive, if you're including that outer membrane. But some of why we talk about that is because some of those features actually cause disease in humans, like the proteins in the outer cell wall of the gram-positives. Okay, they can be toxic, like diphtheria, Streptococcus pyogenes, which causes strep throat. Okay, some of those proteins can actually be pretty toxic. The lipids that are in the mycobacteria, okay, those can also be really harmful. And then, of course, like I was saying, like LPS, those macromolecules in the cell wall and in the outer membrane, they're different. If anything is different than what we have, that's going to generate an immune response in your body, which is generally a good thing, right, when it's something like a bacteria. All right. We just survived all the outer membranes. So when we come back on Thursday, we'll do